Last but not least, now it's time for our closing conference, and it's a pleasure and an honor to introduce our last speaker. I think that it's our last speaker, Professor Dr. Roderic Guigó, Director of the Center for Genomic Regulation, CRG, an international biomedical research institute of excellence located at Barcelona. What science do with research data? I'll give the floor to Dr. Guigó. Thank you.
Well, thanks a lot for the invitation. I'm actually not the director of the Center of Genomic Regulation. I am just the coordinator of one of the 4 programs, the program on bioinformatics and genomics. And I will talk about data in bioinformatics and genomics, and I will talk about. I think I will talk about a change that has happened in research in biology, or I will try to emphasize this change. Maybe say last 15 years. I think until 15 years ago or so, most of the researchers in biology analyze data that they produce in their own labs. And what has happened during these years is that a lot of research in biology, at least in my area, is analyzing data produced somewhere else. So there are many issues regarding the access of this data, the storing of this data, the processing of this data, etc., etc. I actually thank you a lot also for the invitation and for you to be here, and I apologize because you're here to listen to me and I have not been here to listen to you. And so I don't know whether what I'm going to tell you will be of interest to you. I hope that, I know that's the last session, that's Friday, it's Barcelona, it's a beautiful day, so I will try not to be, actually this is our institute next to the beach, so I'll try not to give you for too long, and if you have any question or anything you can interrupt me. So what, as I said, I coordinate this program of bioinformatics and genomics, so I am myself a bioinformatician. My work is in the work of my group is in the development of methods, computational methods to analyze DNA sequences, and what you see here is actually the raw data that we work with. So this is not a metaphor, this is actually the data in which the people in my group are working with. So these are files of this sort of sequences of letters, 4 different letters, A, C, G, T. Right? So these files may contain thousands, millions, hundreds of millions, thousands, billions of letters like that. They correspond to genomes, to what's called the transcriptome, the DNA, to the RNA, etc., etc., etc. And we process this data, we generate data of other data types, but the raw data, the basic data, the beginning of all our research starts with this sort of very simple data type. It's, as I said, just files with sequences of 4 letters. Why is this important? Well, I don't think I need to do a basic introduction to biology, but this is the sequence of the genome. The genome is the set of instructions that define the basic characteristics of the biological characteristics of living beings. The genome is what makes a frog different of a wolf, different of a mushroom, right? So it's not because the wolf has a genome and the frog has something else and the mushroom has something else. The bacteria, the mushroom, the wolf, the frog, ourselves, all have genomes that have exactly this structure. I cannot distinguish whether this is the genome of a bacteria, of a frog, of a lamb, or a pine, or whatever, right? But is in the sequence, is in the number and in the specific sequence of these letters, where this biological, what actually makes a wolf or a frog is encoded. And of course, as you also know, is also in this sequence where the biological differences between the individuals are also encoded. So if people that are here are different one to each other, is because in your genome you have a slight changes in one of these positions where I have an A, maybe some of you has a T and someone else has a C. Is there a pointer or something that I can use? Or of course, this is a symbolic representation. It's not that I have written somewhere in my body this text. What happens is funcionava? No, potser sí. Well, anyway, and this is just a symbolic representation, but is actually each letter represents a different molecule. There are 4 types. These are molecules that are actually polymerized in a very, very long molecule called DNA, which forms the chromosomes that are in each of our cells in our body, and each of our cells of our body have the same sequence, which is the sequence of my genome. So this is this research area in which we work. Bioinformatics is an area which is field, if you want, which is just very recent. This is the percentage of articles in Medline that deal with the term bioinformatics compared to the proportion of articles in Medline that deal with cancer. Probably this is the largest number of articles in Medline. Medline is the database of all the research articles related to medicine and biomedicine and biology are stored. So, so this is cancer. More or less this has been stable since Medline was established in the 50s. About between 5 and 6% of the articles deal with cancer. And as you can see, bioinformatics did not exist before the 90s, and since then there has been an explosion in the field. Right? Still, it's not as large as cancer, but it's like 2% of the articles, which is a lot of articles. And as everything else, I mean, when you see the emergence of something, it's because this doesn't really come from nowhere. So the roots or the seed for the emergence of this something, in this case bioinformatics, were set up quite early. And I just want to briefly go over these historical points because they have some relationship with what the message that I wanted to convey. So if we go to 1950, this is maybe the birth of molecular biology. This is of course arbitrary, but you know this is the point in which the time point in which these two researchers, Watson and Crick, discovered the structure of the DNA, the double helix, and actually this polymeric structure, AC, that we represent as ACGT. And this was actually the first year that this person, which is not as well known, Sanger, was able to develop a method to determine the sequence of the amino acid sequence of the proteins. The function of the DNA is to encode the amino acid sequence of the proteins, and the proteins are the main building blocks of living beings, the structural and functional components of the cells. Maybe if you don't know, but you have to know that Sanger is one of the few scientists that has won the Nobel Prize twice. And actually he won the Nobel Prize the first time for being able to infer the amino acid sequence of a protein. In this case was the insulin from the cows. And this happened more or less at the same time that in a completely separate field of research or of technology or whatever, computers were invented. This is a picture of what is said to be one of the first computers in ENIAC in the university, I think in Pennsylvania was. This is electronic numerical integrator and calculator. But these two disciplines, let's say molecular biology and informatics, remain separated until mid-60s. That was the time in which, thanks to the integrated circuits and the transistors and integrated circuits, computers were becoming more affordable, and computers which initially were only on military institutions and banks and so on, were reaching for the first time the universities and the academic world, and scientists could use computers to do analysis of the data. And this person here, Margaret Hoff, was one of the first people that actually used computers to analyze this biological data. So if we have to name someone as the father of bioinformatics, ironically, the father of bioinformatics is a mother. And I'm saying ironically because bioinformatics is still a discipline which is dominated by males. So what did Margaret Hoff did? Actually did something very, very simple. Since the year in which Sanger sequenced the amino acid sequence of the first protein until mid-60s, other groups have replicated the method of Sanger, and they have sequenced many protein sequences in different labs around the world. So in the mid-60s there were about 300. The sequence of amino acids about 300 proteins. And what Hoff did is simply, and of course we're talking before computers, before internet, before anything. So the sequences of the amino acids were published in scientific articles. What Margaret Dayhoff did was very simple. She went over all articles and he collected and she typed it in this book. In this book, all the known sequences of amino acids. So this was the first collection. This is called Atlas of Protein Sequence and Structure. It was just a book that has 300 sequences of amino acids. And just for you that don't remember biology, amino acids are also polymeric molecules, which are made of the combination of 20 different amino acids that will represent by 20 different letters. It's also a series of sequences of 20 different types of letters. And this book, right, this book was the first database, in a sense, the first database in which the collection of information which has been obtained in many different places in the world was put together. And of course, this actually was great because for the first time, researchers, even though it was on a book, they have the ability to compare different sequences. And you know that scientific knowledge arises when you are able to generalize from individual examples. And through the work of thanks to the fact that we had this information all put together in the same place, we were able to learn a lot about the molecular biology, the basic molecular basis of life or biology. This is just an example of what now it looks obvious to all of us, but what actually, thanks to the work of Margaret Dayhoff, was discovered. So this is the sequence of a protein which is called cytochrome. It's a protein that's involved in respiration, and this is the sequence. It's very probably you can understand. So this is the first row in each of these groups comes from cow. The second row comes from human, and this is from a protist, and this is from a Neurospora crassa, which is a fungi. So and this is the continuation, right? This is the continuation. This is just a fragment. So the first thing that we could realize that all these proteins that have the same function in different organisms, even organisms that were very distant phylogenetically. I'm actually not sure what is the phylogenetic distance between Neurospora crassa and human, but it's probably between 600 and 900 million years. So we separated from a common ancestor almost 1,000 million years, and still the sequence of this protein is actually quite similar, remarkably similar between Neurospora crassa and ourselves. So suggesting that sequences that have the same function, even in very distant organisms, have very similar sequence. Also, the other thing that you could see is that species that were closer in evolution had a more similar sequence than species that were distant in evolution, reflecting that the similarity between the sequences captured our evolutionary history. And of course, by looking at this, and maybe you're able to understand, as you can see, there are positions that are very much conserved through evolution, like this, that hasn't changed in 900 million years, and there are other positions that have varied a lot, right? So the assumption, of course, is that whatever the function of this protein is, this region of the protein that has been conserved over 900 million years is probably more important for the function than this position here that has changed a lot. So the point that I wanted to make is that this was the first compilation of sequence, and thanks to this compilation, there was a jump in our ability to understand life at the molecular level, and this actually, as I said, this triggered the area of bioinformatics, which I think the core of this area of research is just comparisons of text comparisons. So, because the problem that as the number of sequences were growing, the problem was to build these alignments, but this is called an alignment of sequences, in which each position in principle corresponds to the same functional, each column corresponds to the same functional position, to do this in an automatic way. And these were methods of dynamic programming that were fast and were efficient. So with this, I think that this field in which I am working was essentially born, because there were methods that were developed in purpose specifically to address problems that were arising from the analysis of molecular data in biology. The growth of the sequences during the years was exponential, and in the early 80s, the first electronic databases were established in the United States in Los Alamos National Laboratory. It's called GenBank. It's not in Los Alamos National Laboratory anymore. Maybe you know that Los Alamos National Laboratory is the laboratory in which during the Second World War the Manhattan Project was carried out. This is where all the physicists met to develop the atomic bomb because it's in a remote area, etc., etc. Anyway, in this laboratory is where the GenBank database was established in '82, and in Europe, in Heidelberg, in the European Molecular Biology Laboratory. This actually happens more or less at the same time that the personal computers were in the market. It's actually this now for us it looks obvious, but before, right? For instance, when I started my PhD, there was a computer in the University of Barcelona, right? So you had to access to some sort of terminal to a computer. Now this changed a lot the way in which people interact with computers because the computers were finally at the desk of the researchers. So allowing, empowering the researchers the use of computational methods to analyze data. And because the data was growing, and there was the need to look for similarities between sequences, because similarities between sequences, as I said, reflect biological similarities, functional similarities that are important from the biological standpoint. New methods of database searching in sequences were invented, etc., etc. And for instance, in one of the landmarks in the folk history of bioinformatics is this work by Doolittle, in which he was using these recently established databases using his personal computer and some of the methods that had just been developed to compare all the sequences of the proteins against all sequences of the proteins, and he discovered that an oncogene, which is a gene that's involved in cancer, was very similar to a growth factor, a gene that's involved in the growth of the cells and the growth of the tissue. So actually illuminating, I think maybe for the first time, the relationship between cancer and the normal growth of the cells. And this was actually quite a demonstration that the computational analysis of biological data would actually have important implications for understanding biology. So when the Human Genome Project was implemented, when the program was designed, it was already clear. This is in the early 90s, in 1990. It was already clear that computers were going to play a very, very, very important role in research in biology. So this document by the national, I think this is the, well, anyway, I don't know if it's NIH or DOE, because the Human Genome Project was funded by two different agencies in the United States, the NIH, National Institutes of Health, and the DOE, the Department of Energy. So essentially it says, without powerful computers, genome research cannot proceed. So in the 90s, during the 90s and early 2000, this is when the Human Genome Project took place. This was a project that was conceived to last from 1990 to 2005. And as you can see, it was completed ahead of time. Partially because in the late 90s, this was a publicly funded project. But in the late 90s, there was a researcher in the United States, Craig Venter, who actually said that the way in which the public money was being used for this project was waste. The project was badly designed, and that he himself could actually sequence the human genome. And he actually created a company called Celera Genomics, and he was able he claimed that he was able to sequence the human genome, and because there was fear in the let's say in NIH and in the public institutions and in general in the scientific community, that something that in principle was going to be so important as the sequence of the human genome was going to be in private hands. So there was an acceleration of the project. So at the end, actually I don't have it here, but at the end, the two genome projects, two human genome sequences were published simultaneously, one in Nature and the other in Science. And this was in the year 2001. Okay, this is you see it here, Science. This is the where Craig Venter published the human genome sequence, and Nature where the public consortium published the human genome sequence. And I remember that there was already at some times in discussion, because by that time, when you publish an article in which you had some genetic sequence, most journals had as a condition that you deposited the sequence that you have obtained. At that time, it was a sequence of a gene or of two genes, or not of a genome, or of a genome. Eventually, you have to. There was actually a condition that you actually deposited this sequence to a public repository. Craig Venter did not want to do that because, of course, the business model in which his company has been built was into actually mining from the information that apparently was going to be of vital importance, that was going to be encoded in the sequence of the human genome. And this, of course, would have made it possible his paper to be published in Science. So there was a compromise reached in which Craig Venter did not submit the sequence to the standard public repository, to GenBank, but actually he put the sequence in a place in which researchers could still access it. That was a little bit silly because in reality the human genome sequence had no business value, so the company was a failure in that sense. Okay, the expectations about the economical value of the human genome sequence were overly exaggerated because in reality, to mine the human genome sequence for biologically relevant information is very, very, very difficult. So so as you can see, I mean, this explosion of our field, my field, actually occurs during the development of the Human Genome Project, right? Started when the Human Genome Project started, this is where the emergence and the explosion of this field. You could claim, and maybe you're right, that, well, this may not anything in particular to biology, right? Because it's also from the 90s where the use of computers was extended to many different areas of research, economy, meteorology, physics. I mean, all these disciplines generate as much or actually much more data than biology. Therefore, it's nothing in particular on using computers on biology that would, maybe if actually we look at other disciplines that use computational methods, we actually see a very similar trend. However, I did this search yesterday in Medline, in Google, and this is the number of hits that I get when I use the term informatics with a prefix that refers to other disciplines, right? So I think that the table speaks by itself, right? So it's like 2 orders of magnitude. The terms coming from bioinformatics, that coming from any other area that I can manage. Maybe I actually miss something. I don't know. It's possible that there are some other discipline, although I doubt it, that has this termination informatics that had a number of hits which is similar to bioinformatics. But I think that, as I said, the table is quite impressive. As I said, it's not that biology generates more data than astronomy, for instance, or maybe even sociology. I don't know. The fact is that there is something to some extent unique to this field, as reflected by this table. And I don't know what is unique. I had two hypotheses that are compatible, which I would like to share with you. The first one is sort of, let's say, fundamental in the sense that speaks to the nature of the data and nature of biology. So one thing you could say, well, bioinformatics has been so successful because the data in which we work has a strong, the symbolic representation of the data has a strong value. So there is this, of course, this is not my genome. This is a symbolic representation of my genome. But essentially, for all practical purposes, this is my genome, right? So, right, this data, the symbolic representation is very amenable to computational analysis. Computers are very good at dealing with this sort of data, and also because basic biological process can be understood as a computation. This is the first step in the unfolding of the instructions that are encoded into the human genome, right? So I said that my genome determines the color of my eyes, there's my skin, my height, predisposition to some diseases, etc., etc. And what does this mean? Well, how actually this happens? Well, I mean, the first step, as I said, into the unfolding of these instructions is the translation of the sequence of of the DNA, well, actually it's the RNA, but doesn't really matter to the sequence of the protein, right? So a region in my genome, right, according to a very well set of established rules, is translated to the sequence of a protein. Every 3 letters here are translated to a letter here. This is the DNA, this is the protein. So this is, if I have to tell someone what the computation is, I think it's very difficult to find a better example on this. This is exactly what we define as a computation. We have an input, which is the sequence of symbols. We have an output, which is the sequence of symbols, and we have a program, which is the genetic code. So at the basic mechanisms, of biology, of the transference of information from the genome, I think cannot be understood as a computation themselves. So there is this metaphor, which is not only a metaphor because it has practical implications, right? That both the data and the processes are like computations, and therefore, for that reason, bioinformatics has been much more successful than other disciplines that are also using computations. At least as a name, right? As a name. But the other reason, which I think is also important, is openness. So this field of bioinformatics and genomics, I think, has been quite unusually open. Open in terms of the methods that we use, and open about the data that we use. So the early developments of bioinformatics were very much linked to the Unix operating system, and mostly to the free versions of the Unix operating systems by Richard Stallman and the GNU system and the Free Software Foundation. So the idea that software has to be open source, that the best return to the society and to the scientific community is by releasing not only the binaries, the executables, but actually the source code for free, was very much at the roots of the early researchers in bioinformatics, and many tools that we use are actually based on this, on people that are working under this spirit. And actually, I have to tell you, for instance, and I don't think that in my life as a bioinformatician, which started, let's say, in the late 80s, so it's more than maybe 30 years or so, I don't know, maybe 25 or so, I think that I maybe used once a program that I had to buy. I don't think that in my group have any program that we had to buy. So all the programs that we use are open source. In fact, this is a list of the most cited papers in these 20 years in biology. The third most cited paper is a paper that has to do with is a search tool. It's a tool to search in the databases. This is BLAST. This program has always been open source. So I like to compare this with the tools that we use were open source, and the databases that we use were open source, were open access to everybody. I like to compare this to the similarity in chemistry, in cheminformatics, if you want. I mean, the databases of chemical compounds are not public or were not public, and the programs that you use to do the analysis were not public. You had to buy them. And I think that the fact that these things were happening did not promote this area of research. The fact that the tools that you need to work with and the data that you need to work with were all freely accessible, generated a momentum that has made bioinformatics such a successful discipline. So there is a lot of there is an area on this data sharing that is particularly let's say it's slightly more complex. It has to do with genomic information, and I think that I'd really like to review what has been the criteria for releasing this data. So most of the funding for genomic researchers for almost everything comes from the United States, with some contribution from the UK. Therefore, more or less, the principles that the NIH is the National Institutes of Health, established for releasing data are the principles that are using elsewhere. So So this is in '96, in the middle of the Genome Project. The first sequences from the first fragments of the sequences of the human genome were published, and there was the need to establish some rules on how this information was going to be distributed and what were the conditions according to which researchers that were not producers, right? Researchers that were users of the data could use this data. So essentially, and in general, the NIH policy has been the following: has been making the data as soon as possible, available as soon as possible to the scientific community, but at the same time, try to protect, to some extent, the rights of the researchers producing the data to publish the results. So the idea is that if you get funding for a research project that produces data, you have to release this data because this data is funded by the public, and therefore everybody has the right to access this data. But on the other hand, we understand that you are putting a lot of effort in generating this data, and you should have some priority to analyze this data and make a publication. And this is more or less what has been evolving in the NIH policy. So essentially, for you to make an idea, this means that every sequence that was more than 1 KB means 1,000 bases. For you to have an idea, this is the human genome has 3,000 million bases. So this is 1/3 million of the length of the human genome had to be deposited because the human genome cannot be sequenced on a shot, right? It's just a technical thing. It's sequenced in small pieces, and they were released. But it doesn't say anything about conditions for publication or analysis of this. The aim is to make the entire sequence freely available in the public domain for both research and development in order to maximize benefits to society. So when the Human Genome Project was already finished, there was some evolution on these rules. Now, any sequence has to be larger than 2 KB, which is twice, has to be deposited within 24 hours. It's not before a recommendation. Now it has to be deposited within 24 hours. And whole genome assemblies are to be deposited in public nucleotide sequence databases, blah, blah, blah. But it doesn't say anything about publication, whether the users of this data have rights for publication. These rules evolved when let's say one of the largest post-genome projects, which is called ENCODE project, was launched by NIH, actually specifically by this institute. This institute, NIH has many institutes. The institute is National Human Genome Research Institute, right? So here is the following. Resource users are the users that do not produce the data, are asked to respect the ability of the producers to publish an initial analysis of the data, right? But at the same time, NIH is interested in rapid release of the data. So say to facilitate this compromise between unrestricted use of the data and ability of the data until publication, NHGRI will promote, not enforce, observation of enhancement period during which resource users may freely use the encode, mode, encode data to design and carry out their own research programs, but not to submit publications that use unpublished encode, mode, encode data without prior consent. So the idea was that, of course, we have the data, we publish the data, people can use the data, but they cannot publish any results on this data until the data producers have published the first paper, or at least 9 months have passed. The rules have further evolved. This is actually the new NIH genomic sharing data policy, in which actually, if you are funded by the NIH, you should abide to this policy. So the investigators have to submit the data to some designated public repository, and the NIH will release the data and no later than 6 months, and 6 months is the maximum. It could be 2 months, 3 months, whatever. And once the data is released, there is no restrictions. So now there is data producers really don't have essentially very little advantage over data users, for publishing the results of the analysis. Because the truth is that I am involved in some of these projects. The first 6 months is mostly QC, quality control of the data. You discover all the problems, etc., etc., etc. So there is, I think, this I don't know how well I'm doing time, so it's too late. But there is this tendency or this trend towards making the data produced through the genomic data produced through research funded publicly to be released as soon as possible to the public domain, and essentially without any advantage given to those that produce the data. 6 months is really not a lot. I wanted to finish with what I think is going to a problem that we are going to face very, very, very soon, if not, we're actually already facing it. So there has been this since the publication of since the completion of the Human Genome Project, the obtention of the first drafts of the human genome sequence, there has been a revolution in sequencing technologies, and I think that this is just to give you an idea. This is more or less what is the status today. So the Human Genome Project took 14 to 15 years. It took hundreds of scientists working in 5 large centers around the world, used hundreds of machines, and had a estimated cost of $3,000 million. So at that point, like 12 years ago or 13 years ago, it was impossible to sequence a sequence genome, right? $3,000 million is a lot. Today, just 12 years or 13 or 15, let's say 15 years later, we have machines like this that I'm picturing here that can sequence 50 genomes in one day, 50 genomes in a day, and a cost of about $1,000 per genome. I will skip this. I just have this as an example, just for you to have an idea what actually this means in terms of what this means in terms of technological advance, right? So because usually you hear that one of the technologies that had a larger progress is actually computer science, no? And we often hear that, well, if the car industry has progressed as much as the computer industry, now we would have cars that will run 10,000 kilometers with 1 liter of gasoline and a speed of 2,000 kilometers per hour or something, this sort of comparisons, which is telling that actually the computer, the computational technology had actually evolved very, 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 very fast. And I did this very simple calculation, right? So this is the first computer. This computer that had this was actually quite large, right? Had 30 meters for 2.5 meters for 1 meter width, and actually it could compute 385 operations per second. So if you actually normalize by the space and the time, this computer were able to do 10 to the minus 6 operations per second for centimeter cubic. The computer that I usually use is a little bit old, so probably this is much faster now. It's a Mac Air of the 2010. So this computer can make 130. This is the size of this computer. It's tiny, as you know. It can make more than 130 million operations per second. So 10 to the 5 operations per second for square centimeter. How you call centimeter cubic or whatever, not square centimeter cubic. So in 60 years, right? We've seen 11 orders of magnitude of increase in the capacity of the computers, and actually this is more or less, if you do the calculation, it means doubling every 2 years, which is what Moore's law is. I don't want to go into details. These were the machines in this company that sequenced the human genome, the human genome. These are robots that were. I just want to know that there are no people. The sequence of the human genome was obtained by robots, not by people. And this has this capacity of sequence 5, 10 to the 3, 10 to counting the space and whatever and the time. This machine has this capacity. So in only 12 years, right, we have increased 8 orders of magnitude. So the generation of genomic data is much faster than the capacity of the computers to deal with it. And this generates a lot of problems that I will not deal with them, with data compression, etc. The bottom line that I wanted to make is that finally, right, the time has arrived that our genomes will be all sequenced, and I would say that in the next, I may be wrong, but in the next 5 years, everybody in this room, maybe in the next 2 years, maybe 3, everybody in this room will have the genome sequence. I actually had part of my genome in my phone. I have an application here, it's called 23andMe, I can show it to you, where I have the results of my risk for different diseases according to my genome, right, in red or in blue. So this is not science fiction, actually this is a result of the analysis of 1 millionth of my genome, just 1 millionth, one every million bases in my genome has been analyzed, and I get all this information about that, about my predisposition to have several diseases compared to the average predisposition of the general population, right? But that's not the point that I wanted to make. The point that I wanted to make is that this has arrived, and the policies for data sharing that has been in place until now are probably not valid for this, because there is a debate on the value of privacy versus open access. I think that we usually take for granted that privacy is a good value, and I am not so convinced, and I will try to explain you why I am not so convinced, at least in the case of, in the context of genomic information. So far, the few data on individual data, right? When we talk about the human genome, this human genome that was sequenced was some sort of standard human genome, was a mixture of many people. This not corresponds to anybody in particular. It's a reference genome. There is not a human genome, right? Because we are different, because our genomes are different. But there is something which we call the reference, because the human genome is different of the human genome. There is something very common, right? So this is the database. It's called the EGA. Means European Genome. Well, this is the database in Europe. It's called European Genotype Phenotype Archive, and it has all the genomes or genomic information that has been obtained from individuals for research purposes, right? This database, because it has information that can be attached to specific individuals, has to be protected. This information is not anymore freely available, as free as it is the information that's in GenBank and the typical databases that we have been using in our research, because access is controlled by data access committees, right? And this is a large database. In the United States, the name of this database is called DBGAP. It's increasing exponentially. So this is just from 2010 to 2015. It has at this particular moment 1.6 terabytes, I think, or it's 1.6 petabytes of information. Is 1,600 terabytes. It has 200,000 files, etc., and it's growing and growing and growing. So why I am bringing this here? Because this is a database, by the way, which is maintained by the Center of Genomic Regulation in Barcelona, where I work, and the European Bioinformatics Institute near Cambridge. It's a joint venture between these two institutions, and more or less between 40%, 50% of the effort and the funding of this database goes to enforce security and privacy, so that the data is secure, that people cannot get into the database, that only people that have right to access the data access the data. So a large part of the effort in this database is used to maintain security and privacy. So this means that the effort that can be devoted to more basic questions, for instance, I don't want to be demagogy or something like that, but for instance, to try to investigate the relationship between genetic changes, changes in our genome, and phenotypic changes, for instance, diseases, is half, right? Because a large part of the amount of resources is limited, and if half of the resources are limited to security and confidentiality and privacy, means that to do basic research or to have less resources available. This is why there is a movement led by George Church in the United States, which actually claims that having this data private is counterproductive. It doesn't help anybody. It doesn't help individual people. It doesn't help the community. It doesn't help the society. And he started in 2005 this project, and it's dedicated to creating public genomic health and trade data. Sharing data is crucial to scientific progress. And says, donating your genome and health data to science is a good way to a diverse person to become a public resource. So the idea is that people participated in this project. They put their genome and their health data, right? Their health data on the public domain. So for everybody to look at it, it's not restricted to anybody. So researchers can look at the data, lawyers can look at the data, insurance companies can look at the data, your government can look at the data. Because the thinking in this group of people, in Church and others, is that actually privacy doesn't really help. It's actually counterproductive in this particular case. Today, if you are Catalan or you follow the Catalan press, there was I have to admit that this is a very it's a topic that is actually quite. There is a lot of debate regarding this topic in society, which I think is good. In Catalonia, there was this plan to release medical information, all medical information that's on the public health system for research purposes, but because of political pressure from a number of parties, this plan was sort of put on hold, and what yesterday happened is that a number of scientists, including myself, we actually signed a manifesto in which we were asking that this project is on the benefit of the society, that not making this data available for research purposes would be a responsibility, and we actually will be the managing for the society. And I'm just bringing this here because I think an issue that will be really very important, and I think will make us to reconsider in general what is the value of the privacy regarding our data, is the access to the genomic data. And I think that I want to finish with this message. We really need to assess what is the cost of privacy. It's not for free. If we want to have something secure, we have to pay. If we want to put an alarm in our house, because to avoid the burglars to enter, we have to pay. And I guess that we're willing to pay if this is sensible, but maybe the price that we have to pay for privacy in other components of our lives is too high. Genomes in isolation have little value. It's only through the comparison of the genomes of hundreds of thousands of millions of people that we can actually infer relationships between changes in the genome and phenotypic changes, in particular diseases or predisposition to diseases, and also genomic privacy. And I think this is quite important, is an illusion. There is no way in which I can guarantee that my genome will be made public. And this is different to other kinds of data. The most dramatic case is if I had an identical twin, I actually share my genome identical with my twin. I may decide not to release my genome, but if my twin releases his genome, because in this case it will be his genome, my genome is released. And for instance, I may decide not to release my genome, but my children may decide to release their genome. And my children share half of their genome with my genome. Therefore, my genome will be, to some extent, released as well. So there is a limitation, an intrinsic limitation in the case of genomic information to what can be private and the control that I have over something that I think belongs to me. And I think that we need to reconsider this idea that it's sort of common that my genome belongs to me, because it actually is not the case. And well, I'd like to thank you for your attention. Because I discussed this before, I just wanted to to comment on. I wanted to talk about something else, but I did not have time to prepare it. But someone said that this could be interesting for your audience. Is there is a trend now in our area in which a lot of the scientific discussion and analysis is moving beyond traditional means of communication, like journals, to other more modern. I mean, I'm talking to people like me that don't have Twitter and Facebook and all these things, and that's why I call modern, because it's modern for me. Maybe it's not so modern, like Twitter, Facebook, and all these things. So there is a lot of now of discussion that is being moved, a scientific discussion that is being moved to blogs and Twitter. And actually, this is an article that appeared in Nature last week, which actually was commenting on the fact that there was this tweet. This is a Twitter blog in which scientists, Gilad, which is very well-known scientist, published the analysis of a paper that has been published in PNAS for another respected scientist, Mike Snyder, saying that it was wrong. And the results of the analysis were in Twitter. Right, so I don't want to talk more about this just because I didn't have time to prepare it. I actually wanted to, but I didn't have time. But I just wanted to mention it because I've been told that this could be of interest to the audience here. So that the traditional means of scientific communication through journals is now, let's say, being expanded to other less orthodox means of communication. And with this, I want to stop. Thank you.